think we've got a good number of people in the audience here, so maybe I'll just jump in the interest of giving our presenters as much time as they would like to run through their stuff and also leave time for Q&A at the end. So thank you for joining us for today's Collaborative Science for Estuaries webinar series. This is Nick Sobral with the Science Collaborative. Today's webinar, which is the first of 2026, we're kicking off with a team from California that developed a shellfish biosensor monitoring system to complement existing long-term water quality monitoring and provide real-time feedback on estuary conditions. You'll hear more from the team about that in a few minutes because they know way more about it than I could ever know. And I'm looking forward to this one as well because I actually am not as familiar with this project. So this will be fun for me too. Before we get into that stuff, I just have some quick housekeeping things to run through and a quick introduction to the reserve system, this webinar series, and this program. So, the National Estuary and Research Reserve System is a national network of 30 unique reserves, as shown on this map. This is a NOAA program that works in collaboration with a local place-based partner, either a state agency, university, or nonprofit at each site. Each reserve site includes programs focused on the activities that you can see on the slide here, which include land stewardship, research and scientific monitoring, training, and engagement programs for the public and local officials, and education. The Science Collaborative's mission is to support collaborative research that addresses coastal management issues that are important to the reserve system and that strengthen the system's ability to impact coastal management and decision making. And one important way we meet this goal is by sharing these collaborative science accomplishments publicly through a variety of channels, including this webinar series, which is the Collaborative Science for Estuary series. This series features project teams supported by the Science Collaborative, program staff, and partners. Quick note on your contributions. So webinars are one way that we share important project impacts more broadly, and they're also a really valuable learning opportunity for us as a program. Insights from speakers and participants like you are really valuable in helping us inform how we support others in doing their own collaborative science work, for example, in the development of future workshops and webinars, or in resources such as tools, tips, and case studies that make their way into our guide for collaborative science, which is open access, and you can find it on our site. With that in mind, we just want to inform folks that any insights or feedback that you share, for example, in chat or via the exit survey that launches after you close the session, may be used for program learning and development. Please note that if we capture a direct quote from you that you'd like to use for learning purposes, we will reach out to you beforehand to seek permission and provide an explanation of its use. And if we use any quotes, they would be used anonymously. All right, housekeeping stuff. So all attendees are muted upon entry, as you probably noticed. We'll be handling presentation-related questions via the Q&A feature. You can enter questions as they occur to you throughout the session using the Q&A feature if you'd like. We'll plan to leave time at the end to answer as many as we can. You'll also be able to verbalize questions at the end of the Q&A session if you'd like to, so just raise your hand if you'd like to verbalize your question, and I'll take you off mute, and you can come off mute and ask your question verbally. Chat, as I mentioned at the beginning, is visible to all, so please use it to talk amongst yourselves. And you can also use it to ask the organizers, which are myself and Ariana, any technical questions if you have technical questions as well. And with that, I will introduce our speakers for today's session. So joining me today are Luke Miller, an associate professor of biology at San Diego State University and served as the project lead for this project. Kristen Goodrich is the director of training and engagement at the Tijuana River National Estuary and Research Reserve and served as the collaborative lead for the project. And Kevin Marquez Johnson is an aquaculture extension specialist with the California Sea Grant. And I believe I'm passing it to Luke now, so I will stop sharing and Luke can take over.
All right. So thank you, Nick, for the introduction, and thank you to Kristen and Kevin as well for being here for this talk. I'm just going to very briefly introduce the project and what we were trying to do, and then we'll hand it over to Kristen and then Kevin, and then I will come back at the end to talk a bit about what we accomplished. So as far as the project that we're talking about today, it was funded by the Science Collaborative, and our team included myself, Dr. Sarah Giddings at the Scripps Institution of Oceanography, and Dr. Kristen Goodrich. And our sort of primary end user was targeted at the Tijuana NERR, and Dr. Jeff Crooks there, who is sort of help develop a lot of their needs there. And so we tried to tailor a project that was tailored to one of their particular needs, where they have of course, in place long-term environmental monitoring, water quality monitoring through the system-wide monitoring program or SWAMP program, but had the sort of need or desire to try to augment that existing physical water quality measurement with and monitoring with something that sort of gave them a real-time biological feedback as well. And so potentially using something like oysters or mussels as a sort of live to the internet kind of biosentinel system. So our goal was to try to develop and implement a sensor system that we could attach to live animals, put them out in the estuary or in the bay in the various places that we were working, and actually be constantly recording both their shell opening and closing, but also another physiological measurement like heart rate and also then potentially broadcasting at least that shell opening and closing data to a real-time website. So that brings us to our sort of proper introduction to the project goals here by Kristen Goodrich. So I'll hand it over to you, Kristen.
Thanks so much, Luke. And hi, everyone. It's wonderful to be here and get to share a little bit about a project that is very close to our hearts, the Habitat Heartbeats project. I think in the spirit of collaborative science and, of course, the types of projects that we pursue through the Science Collaborative, I was hoping today to share a little bit with you all about some of the inspiration for the Habitat Heartbeats project, but also specifically how it directly informs many of our management needs, both at the Tijuana River National Estuary and Research Reserve, but also for many of our sister estuaries in Southern California. And through the project, what we came to learn were that there are many other needs that we believe and have evidence that this technology can help respond to. So I'll start just with a little bit of context on where we are here in Southern California and a bit about the estuaries that are very special and rare here. We've lost about 80 to 90% of our coastal wetlands in Southern California. So the ones we have are incredibly important, and being sure that they are as healthy as they can be is a priority of ours at the reserve and among many of our partners here who manage coastal wetlands. So of course, estuaries everywhere are ecologically complex, but here in Southern California, we can say the same. Our low inflow estuaries in California experience large swings in water quality during episodic rain events. We also encounter regional pollution in many of these urbanized centers that the estuaries are adjacent to. Of course, rainfall intensity is changing with the climate, and we're also experiencing major ecological events on a very short time scale. But here in Southern California, winter waves can really be problematic for us. They can lead to mouth closures, which directly affect periodic inlet closures, affecting salinity and dissolved oxygen. The photo to the left is one that was featured in our local newspaper, the San Diego Union Tribune, but depicted a pretty dramatic event in 2016 where the Tijuana River mouth closed, reopened after some flooding, but there was extensive die-off of leopard sharks. And we saw conditions change rapidly because of that mouth closure. We are very actively monitoring for that through, as Luke mentioned, our system-wide monitoring program and protocol, but we are very interested in figuring out if there are ways in which we can better both predict and real-time respond to events like that. On the right, a photo from Los Penasquitos Lagoon, another estuarine system in our region that also is affected by these mouth closures and often has to open the mouth to be able to really make sure that those oxygen conditions stay stable. So I will just say that the Tijuana River Estuary has a very unique challenge in that being highly urbanized and three-quarters of our watershed being in Mexico, we are a transboundary and multinational wetland and watershed. And so the inputs from this highly urbanized setting, the estuary surrounded by the city of Tijuana, the city of San Diego, and city of Imperial Beach, we face significant pollution challenges. And so for us, being able to discover, experiment, and apply new technologies and tools to help us better monitor, respond, and make decisions about water quality are of great interest to us. So as Luke mentioned, when we were approached with this potential to collaborate and think about this technology directly meeting some of our very immediate management needs, we were very excited. So just specifically, some of the management needs at the Tijuana River National Estuary and Research Reserve, again, really are connected to this complex, highly variable environment that is often punctuated by extreme ecological events on a relatively short time scale. We, of course, measure physical variables like temperature, salinity, oxygen, but often we're inferring the health of species without living input. Again, another really exciting opportunity for us to be able to collaborate with Luke and partners on experimenting with these biosentinels. So a little bit of context too, and historical context. Here in Southern California, shellfish have taught us about how the area has changed. The fossil record has revealed species abundance and habitat changes. And indigenous communities, naturalists, hobbyists have collected shells for thousands of years. Now we have the opportunity to monitor them in real time to see short-term scale changes in the environment. So Luke mentioned Dr. Crooks, our research coordinator here at the reserve, who was very instrumental in helping to inform us and the project really about very immediate and specific user needs and management needs at the reserve. But this is something he shared with us in one of our many conversations about how to develop the project to be responsive to those needs. But you can read here, he shared with us that the critters living in these systems are seeing things differently than us on really small spatial scales in really complex systems. On the bottom here is a photo of some of us at one of our annual meetings. And this one, I believe, in Wells, Maine, where we were able to present some of what we were learning real time with the NERRS community and later inform some future collaboration opportunities and partnerships with folks at Wells who are experimenting and looking at this type of technology using lobsters. So I know Luke will get into this in great detail, but for us, again, this exciting opportunity of utilizing bivalves to inform management. One, we can get direct feedback on ecosystem health to link the biology to the physical and chemical primers that we've been logging for nearly 30 years here at the reserve. And we also see it as a less intrusive and more intuitive mechanism or method for getting this data real time. And I think I think I'm ending on this slide, but it's one that but I'm really interested in, and what kind of drove my interest in involvement in this project as a social scientist was really trying to understand user needs beyond just those at the reserve, which are of course very real and almost second nature to us. So what we did in the pursuit of really understanding various and diverse user needs as part of this project was conduct interviews. And we did that with various fields and various sectors. And these are just a sampling of some of the kinds of questions that came up in response to the questions we asked about biosentinels and possible opportunities for application of this technology. So here you can see some just samplings. Folks were interested in how biosentinels can help us understand where to put native oyster beds, for example, in living shoreline restoration projects, some of which are happening right now here in San Diego Bay. Many folks were interested in are there other organisms that we can learn from? We know that yes, some partners in Wells, our friends in Wells are working on this actively right now. Another group of folks, and this is what I'm really excited to hear from Kevin about, is the aquaculture sector and field. Folks are wondering about how real-time data from these biosentinels can help us make decisions about aquaculture operations itself. So Kevin will be up next and share a little bit about his perspective on that. And this, I think this this one quote here, can this complement existing monitoring programs to provide a better understanding of estuary dynamics and guide decision making? Was one directly here from our reserve and our reserve partners with California State Parks and U.S. Fish and Wildlife Service, who are really intrigued about the opportunity for utilizing biosentinels as a way to, in real time, have decision making be informed by these organisms. And then lastly, but certainly really important and something that I was really in awe of during the project, Luke's involvement with students both at the university, but in the public as well, and K through 12, showing students some of these what we lovingly started to call in the project oyster backpacks, how the students could really see these organisms and this technology real time to help them learn about water quality ended up being a really amazing outcome of this project overall. So this is just a sampling of the kinds of feedback we heard along the way, mostly in the forms of questions and curiosity among the end user community that spanned both estuary and coastal managers to aquaculture industry to those folks who are involved in establishing living shorelines and restoration projects. So with that, I'm excited to hand it over to Kevin, who I believe will be sharing a bit with us a deeper dive in sharing some perspectives on how the aquaculture community engages with this technology, a little bit about just the aquaculture in California overall and some opportunities that he sees in potential for utilizing this in the field. So thanks for the opportunity to share a little bit about our management needs at the Tijuana River Reserve.
Hi, thank you. Yeah, I'm excited to be here and be part of this conversation. I've been following these oyster backpacks for quite a few years now, and it's really exciting technology. And I hope to show you a little bit how it might have some application within aquaculture settings. Um, but before we get there, I am an aquaculture extension specialist with California Sea Grant. And as such, I wanted to make sure that folks on this call knew a little bit about aquaculture in California. It's not the biggest industry as you compare aquaculture to, like, say, East Coast or the state of Washington. But we have some really fun things going on. So what is California aquaculture? California is mainly focusing here on the marine aquaculture component, has an economic impact of about $57 million, again, which pales to other industries. Um, but this industry is really driven by the production of shellfish and seaweeds that are relatively low trophic level organisms that are being sustainably produced throughout our state. Um, Producing these shellfish, these low trophic protein sources, is a really important way as we think about food security moving forward, um, as our environments are changing. And one of the things that I love about my job is the opportunity to work with farmers, shellfish growers who have a really deep respect for their environment and really care for keeping it clean. Keeping a clean environment keeps a healthy oyster, which survives and hopefully provides some income for them. Oh, I talked about this a little bit. So in California, most of the marine aquaculture is either land-based, and so there we're seeing things like abalone and seaweeds being produced in land-based. Tidal lands where we're seeing our clams and our oysters being produced, and then open water where we're seeing some mussel and also oyster production occurring. And potentially in the near future, we'll see expansion of mussel farming using the NOAA's aquaculture opportunity areas off of Southern California. Um, California, though, we consume here in the state about 280,000 tons of seafood, but we produce less than 90,000 tons, and that's a combination of aquaculture and fisheries production numbers. We're importing hundreds of tons of seafood into the state for our own consumption, really calling for us to really think deeply about the seafood that we're eating, where it's coming from, and the sustainability of that consumption. Here in California, we have 7 primary oyster growing locations across the state. And these are these black circles here, from Humboldt all the way down to San Diego. Across all of these areas, the shellfish farmers lease about 5,740 acres, which sounds like a lot of space, but in actuality, they only farm a small bit of it. Less than 500 acres is actually actively farmed, about 8.5%. And what that leads to is that these growers actually end up being stewards of large sections of our estuaries. And here in where I'm based in San Luis Obispo and Morro Bay, when we had some eelgrass die-offs in the estuary, it was actually the shellfish growers who were one of the first ones to come to the table with the Morro Bay National Estuary Program and help with a lot of that restoration using their equipment and their knowledge of the estuary to help get eelgrass restoration out and going. Because again, the health and the wellness of these water bodies is critically important to their livelihoods. And so they invest a lot of their time and effort to keeping these water bays clean. Just a bit on the economics. Here in California, we have kind of a boom and bust scenario, and this is pretty common for a lot of aquaculture, and there's some missing data in here as well. But overall, we see California can produce about 800 tons of oysters, worth about 16.5 million. And the great thing about this is that over 60% of this goes straight to consumers. So these are oysters that are being produced in the state and being sold straight to restaurants and to consumers directly, and not going to big processors. But despite this large number, we still import a significant amount of oysters. So we produce 800 tons, and next bullet point will show us that we actually import 33,000 tons of oysters worth about $300 million into the state. And those are importations into California major ports. So some of those are going elsewhere, but it just shows the need for oyster production. And what we see in this graph is these ups and downs is not a change in growers' attempts to produce. It's more related to oysters not thriving and having mortality events. And our next slide will talk a little bit about that. But the consistency of production changes for these growers based off of environmental conditions. Just over these last 3 years, I've been part of a number of studies tracking oyster growth and mortality throughout the state of California. And over the last 3 years, we've observed sometimes more than 75% mortality on farms across the state. And we really don't understand a lot of what's driving that mortality. We are able to send samples off for pathology analysis, and we can look, and we really don't see any strong metrics suggesting that the oysters are diseased or have any really health issues. They're perfectly fine for human consumption, but we're ending up with these oysters that you can see in this picture that are about an inch and a half long, that are having these really strong mortality events. And the industry has come to me to actually ask to see if I can help understand what's going on with these. And that's where I first heard about these oyster backpacks, because I think it really provides us some really new opportunities to think about what's impacting oyster health. Right now, the industry and myself, what we have at our hands is the environmental data being collected by the Central and Northern California Ocean Observing System, which is another federal funding organization I use. And from that, we can see some signatures. And so we'll see times where we have warmer temperatures and low dissolved oxygen. We think there might be something happening there. But the oxygen's really not that low compared to what we would see in the literature. And so we need to understand more about what's happening with these oysters, and actually being able to ask the oysters themselves what's going on. It would be an invaluable tool for the growers to be able to have these sentinels with backpacks on their farm and be able to start to track, like, oh, actually we don't know why, but the oysters are all closed up and they're not actually actively feeding. We might have something going on. Having that kind of information is actually really, really valuable to a farmer. They have have things at their disposal. They can move oysters on their farm. So if an area of their farm is showing oysters that are closed up, they could easily move those oysters out of that area and stop farming that area as intensely. They could do an early harvest and bring oysters in and put them in wet storage, and then be able to withstand a mortality event that's happening. But right now they don't have that information, and we're basically seeing these mortality events after they've occurred. And I see the chat. No, we actually we've tested for a lot of different all the diseases that we can think of. There's no disease present with these oysters, with these die-offs that we're aware of, that we're able to identify. And we've thrown some sequencing to look at viral infections and things like that. And it's an interesting one. And so, yeah, as I said, this hindcast data alone is not enough, because all of this IUCE data, the observing oyster mortalities, it's always after the fact, right? So we come out and we're saying, okay, we measured these oysters and monitored them 3 weeks ago. Now we see 75% of them died. When did that happen? We have to look back and we can say, okay, well, we've got this kind of stretch between June and July where we see these patterns happening in the environment, but we can't actually trigger at what point did these oysters start to perform poorly. And so I'm super excited about things like these oyster backpacks and some of the technology that Luke has been developing in his lab, with being able to actually put those on cell phone towers and get data out at a relatively real-time kind of frequency, so that if this technology and being able to be developed in an area like a NERR really provides a really great test bed for aquaculture industry to look at and say, hey, that's something we could probably adopt. And as was said earlier, this technology has really cool implications for aquaculture, for growers to better understand what's happening, and also for restoration. To better understand, just like an oyster grower, is there areas where we shouldn't be putting these oysters? Yeah. And so, as I said, yeah, this technology being developed through this, really valuable. I'm excited to see where it goes, and hopefully getting my hands on a handful of these backpacks myself to play with at some point. And with that, I think that's all I had. So thank you. If you're interested in California Sea Grant, we're on all the social medias. So find us, follow us, and thank you for your time.
Thank you, Kevin. So I'll talk now a bit about what it was we've actually been doing with these oyster backpacks and mussel backpacks down here in Southern California. And I'll just start by introducing you to some of the sites we've been using. I've got three of them outlined here. The first that I'll talk about in a second is our site in San Diego Bay. And so San Diego Bay is a large deep water port. It was originally the outflow for the San Diego River, although the locals rerouted the river at some time in the late 1800s. And so it's got a few sort of smaller river inputs these days, or maybe you'd call them creeks at this point. But it represents now a relatively stable, primarily ocean-driven habitat. And we were making use of an aquaculture facility there in San Diego Bay, managed by San Diego Bay Aquaculture. They have a floating upwelling platform that you see in the picture on the left here. And so we were, for a number of years, hanging oysters and mussels off of this platform and using it as our sort of quote unquote good site, our stable site that is really sort of oceanic in nature. A second site that we used is up in Los Penasquitos Lagoon, which Kristen mentioned earlier. The picture on the right, on the left here, the orange star represents where our measurement and placement was. It's a bit down from the actual mouth of the estuary itself, which is off to the left here. And that estuary mouth is constrained by a highway bridge, and so it really doesn't have the ability to move and widen and so forth. And so it does tend to have flow issues and ocean exchange issues. And Los Penasquitos Lagoon pretty typically closes up at least once per winter, every single year. And the California state parks, who manage that stretch of coast, have to often make the decision about whether they need to bring in big, heavy equipment and reopen that mouth, or whether they can wait and let sort of natural freshwater flows flush the lagoon out. And so that's a big money question for them. A big chunk of a budget every year is to decide whether they need to do that on an emergency basis. They will also come in yearly in the spring and usually close up that mouth and do some rerouting of the channel just inland of the highway there, and try and sort of make sure there's good oceanic exchange. And then our third site that we'll talk about is the Tijuana River Estuary, the NERR site there. And again, our orange star here marking where we were measuring, just north of the mouth of the Tijuana River. And the Tijuana River doesn't close with the same regularity that Los Penasquitos does, but every couple of years or decade or so, the Tijuana River will actually have waves building up sand at the mouth and closing it off, again, like Kristen mentioned in that newspaper article earlier. And of course, the other big thing that Kristen mentioned is the large amount of people living on both the north and south boundaries of the Tijuana River. And so the Tijuana River has had a lot of sort of human pollution impacts that are ongoing. So I want to show you a bit about what we've been sticking on these animals. And I'll say that, bivalves are really, really handy in that they have this outer protective shell that's, good armoring for the animal itself. It's also a great place to glue stuff too. And so, going back to certainly the early 90s, folks have been putting little sensors of some sort on the outside of clams and mussels and scallops and oysters. And we're certainly not the first to do this, but we've sort of developed our own little relatively cheap sensors that we can put on these oysters. One of those is pictured on the left there, and the technology here is a magnetic sensor, a Hall effect sensor. And so we can glue the sensor to one of the two valves of the bivalve, and then glue a little magnet, a little waterproofed magnet to the other valve. And then as that bivalve opens and closes its shells, that magnet gets closer or further away from the Hall effect sensor, and we can measure that as a changing voltage. In a slightly different way, we're also measuring heart rate of these things. So again, on the left is a picture of the little sensor board that we've developed in the lab, and it's using a chip that is the same sort of sensing chip that is in an Apple Watch or a Fitbit or so forth, that is used to measure your pulse on your wrist. And so it is emitting, in our case, an infrared light signal, and shining that through the shell of the animal and hitting the viscera inside. And as the viscera, hopefully you find the heart, is moving, it's changing the reflected light coming back out of the shell and being picked up by that same sensor chip. And so in that way, on the right-hand side there on the computer screen, you can see a little trace, pulse trace, just like you would get off of one of the, again, an Apple Watch or the little clips they put on your finger at the doctor's office these days. Again, it's the same sort of technology. And we benefit from the fact that the calcium carbonate shell of your typical bivalve is actually reasonably translucent or transparent even to infrared light in these near infrared wavelengths. And so we can, again, stick something on the outside of the shell, not have to do any surgery to the animal itself, compared to old ways of implanting electrodes into the shell. And so the animal essentially doesn't experience any major hardship beyond the fact that it is now tethered to this cable. Both of those types of sensors were then feeding the live voltages or signal back to a data logger. For those gape sensors, the opening and closing sensors, we feed those to this Enviro DIY Mayfly, which was developed by the Stroud Water Center back east, for mostly monitoring physical water quality instrumentation, measuring salinity and measuring temperature and dissolved oxygen and so forth. We've built a little adapter board for it there in purple that we can plug our Hall effect sensors into, and then using the this, both the hardware that the Stroud Water Center has developed, and also this Monitor My Watershed website that they helped develop, we can stream over the cell network live readings from individual animals opening and closing onto the website. And so the picture on the far right here shows a trace over the course of about 3 days of the voltage coming out of that Hall effect sensor, where a low value on that screen represents the shell being closed up, and a high value represents the shell being open. In the field, what we end up doing is putting all of that equipment, all of the data logger equipment and batteries and solar panels into watertight boxes, and either hanging them off the side of a dock, or in some cases putting them on a floating mooring here. So on the left here, we've got two waterproof boxes with two sets of data loggers in them, a bunch of cables running out of those, and then running down the mooring line. So on the right, we've got both on the on the right-hand picture, there's the data logger on the left for the heart rate data. On the right-hand side is that Enviro DIY Mayfly that does the cellular broadcasting to the internet for the GAPE data. We've got a little 5-watt solar panel, which is sufficient to keep these things powered so long as it gets to recharge the little rechargeable battery each day. So these are self-contained and can effectively run for months at a time, really just subject to equipment failure due to water intrusion being the main problem. And usually that's water intrusion down at the sensor that is attached to the animal. So we will co-locate these moorings with other standard water quality sensors. So things like conductivity, temperature, and depth, a CTD sensor, dissolved oxygen sensors as well. And in our estuary sites, we're also then placing these close to the existing swamp sensor arrays, the system-wide monitoring program arrays, so that we can also sort of, again, leverage some of those data that have been collected for getting on 30 years at this point. On the left-hand side here, you've got the underside of our mooring, individual oysters here attached. So these ones are floating right at the surface. They are submerged in whatever the surface water is, along with oxygen sensors and a CTD that will be placed just below that. On the right-hand picture there, we're putting a lower rack that sits about 50 centimeters above the benthos, again with individual animals attached and oxygen sensors, CTD sensors, and so forth. And so trying to get an idea of how these animals are responding either at the surface or down deep with the differing sets of water quality changes that can happen in those two different locations. So I'll show you some example data here from our San Diego Bay site. So again, this was our really kind of very stable, very nice oceanic sort of system here. I'm going to run from top to bottom through these different graphs. And what we're looking at here is effectively an entire year's worth of data right now. We'll zoom in on some of this. But at the top, I'm showing you rainfall, temperature in red there. Dissolved oxygen is that middle graph. And then the bottom two graphs are those biological data. The first, the gape, and that is the shell gape, the amount of opening expressed as a percentage between 0 and 100%, where 0 means the animal is closed up as tight as it can be. 100% means that's as wide as we ever see it. And so we've rescaled our raw data onto that common measurement scale. And then the bottom graph is the heart rate. And so this is just like your heart rate expressed as beats per minute. Basically, it is the pulse of these animals. And individual colors in those bottom two graphs represent data from individual animals. And in this case, I'm showing you a mixture of mussel data and also oyster data. Let's zoom in on a little rainy period here in San Diego Bay. So I've highlighted in the black box the region that we're going to zoom in on. So now we're just looking at a couple of days in January, where at the top we've got minor rain events. The corresponding temperature and dissolved oxygen are really nice and steady. These are animals that in this case are submerged about 30 centimeters below the surface, and so a little bit of rain falling on the surface is probably not going to affect the salinity that they're in, and it's not going to radically change the water quality. We see over the course of these particular days and nights, where nights are the darker background and days are the lighter background, we see individual animals in our second to bottom graph here closing up. So that gape value going very low in that case, closing up for an hour or two or three, usually in the middle of the night or in the very early morning. And then for the remainder of the day, sitting there basically wide open or as wide as they want to be. The bottom graph, the heart rate data, shows that for the most part, they exhibit relatively stable heart rates as well. There are occasional dips that you see that often are corresponding to periods when the shells are closed, and we'll talk more about that pattern in some of the future data graphs that I'll show you. But again, this is sort of showing us a relatively stable, relatively benign environment for these animals. And I guess I'll reiterate here the fact that we were able to keep these sensors on these animals and keep these things going for pretty long periods of time. So we've got the entire year of 2024 represented here. We continued this into 2025 as well. We occasionally have to swap animals out if they die or if their sensors die. But for the most part, we're able to collect quite a lot of data over the course of a year. So let's turn from our big deep water port to one of our small low inflow estuaries, Los Penasquitos Lagoon, again up in the northern part of San Diego. So here we've got a much smaller volume estuary. It's really going to be impacted by some urban runoff sort of year round, but it's those big episodic rain events that can really change the character of the water in the estuary over very short periods of time. And then we've also talked about mouth closure events as well. So here again, I've got the same sort of ordering of data from top to bottom and covering a deployment of about 9 months in this case. And so Los Penasquitos Lagoon is a little bit more dynamic in terms of water quality conditions than we saw in San Diego Bay. So you do see some, change in temperature through the course of the year. We see some drops in salinity due to rain events that correspond to the rain events we see up in the very top graph. In green, there, the oxygen graph, we see quite a lot of variations. Sort of when we zoom in, you'll see some of that is day to night. It is also associated with these freshwater flow events that come through. But again, looking at the data on the bottom, the gape data and the heart rate data, for the most part, you see these animals with relatively high gape values. They're spending a lot of their time open and occasionally closing. So let's again zoom in to a couple of days and kind of highlight what's happening there. So here, a dry period where there is no rainfall happening, we get a bit of day to night fluctuation in water temperature that's also going to be associated with the inflow and outflow of ocean tides into this small lagoon. We've got a little bit of salinity change, again, probably tied to both that urban drool that might be running across the surface of the lagoon, but also, again, the inflow and outflow of high and low tides. In terms of oxygen, you see some movement of oxygen here, but in this little highlighted area, it's always staying quite high. 6 to 8 milligrams per liter seems to be perfectly fine for most of these animals. And in terms of that gape behavior in the second to bottom graph, we see again animals mostly open, but we do see these individual nighttime closures. And what I want to point out here is that, again, like we saw in San Diego Bay, those nighttime closures are kind of idiosyncratic. Individual animals seem to be closing for a period of time, not the same time necessarily as their neighbors that are literally sitting in the same water a few centimeters away from them. So there seems to be some, personality, if you want to call it that, among these individual animals that I haven't attempted to try to suss out what drives different personalities here. And then that very bottom graph, again, heart rates relatively stable during this, what I would call relatively benign period of time. Let's highlight a different period of time here in basically late March of 2025, still here in Los Penasquitos Lagoon. And what we're looking at here is, again, a relatively dry period, but we are having these events where in the GAPE plot, the second to bottom plot here, you'll see for everybody is closing up completely for several hours over the course of the night, and then suddenly all sort of reopening in unison sometime in the morning, and going and staying at 100% opening. During those closure periods, you also notice at the bottom graph, the heart rate tends to either drop very low or for us kind of disappear. It becomes so subtle, or we lose the signal enough that we basically can't assign a heart rate. And so during these closure periods, these animals are presumably using up some of their or all of their internal oxygen stores because they're not really exchanging or pumping water with the surrounding environment anymore, and are presumably transitioning over to a primarily anaerobic set of metabolic processes, which are going to be less efficient for them, less energy efficient, and are also not necessarily sustainable over long periods of time, meaning days in this case. So we definitely see some shifts in the way that these animals behave. We saw sort of idiosyncratic individual closures in the previous graph. Here we see some evidence of group responses to something in the environment. And the fact is, I can't necessarily point to what is the change that is happening here. What I don't have to show you, unfortunately, is what the actual surface salinity was, because that sensor managed to fail. And so I'm showing you bottom salinity, whereas these animals we're looking at were at the surface. And it's possible that there was a daily flow of fresh water coming through overnight that was causing these animals to go ahead and close up. But that's speculation at this point. So then let's talk about the Tijuana River Estuary, which is really sort of the centerpiece of our project. Our location, again, sort of a bit north of the mouth, but just off of the main channel flowing out of the Tijuana Estuary. And I want to back up and talk a little bit about the recent, let's say, history of the Tijuana River. The International Boundary and Water Commission has been recording flow rate, cubic meters per second, upstream of our site for about 20 years at this point. And what I'm showing you in this graph here is a whole lot of yellow and basically blue. The yellow areas here represent periods where on that particular day or at that particular hour, there was zero recorded flow in that upstream weir. Any gray background areas with blue lines in them represent periods where the river is flowing. And what I want you to notice is going back to the early 2000s and then moving to the right along this graph, you see less and less and less of those yellow periods, those no flow periods, as we get to around 2022 or so and continuing through the end of 2025. So let's highlight that a little bit. Starting here, 2022, 2023, high flows during the winter time, which we kind of expect when there's rain events happening, but there is some continuous low level of water flowing down through the river all year long at this point in 2024 and 2025. And I think we're sort of seeing evidence of the detrimental effects of that continuous, what is urban flow coming through that river. Alongside that river flow, you often see these drops in oxygen. So I'll just sort of highlight some summertime oxygen data going back to 2008 in this case, taken from that system-wide monitoring program site where we were deploying our animals. So during a typical dry August in 2008, we did see some daily drops in oxygen. As we move forward in time, through 2014, 2019. 2019, we're starting to see even lower measures of oxygen during certain periods. And then 2024 in that bottom graph, when the river is just flowing all the time, now we're seeing daily periods where we can't basically measure anything useful in terms of oxygen in that water. It spikes back up when the high tide flushes some ocean water back into that area. But during low tide periods, when the river is dominating, the oxygen is really disappearing. And so I'll just show you some data from our Tijuana River mooring. Up at the top, we've got river flow rate in place of rainfall, but these sort of higher flow rates here do represent rain events during March of 2024 in this case. During those rain events around March 31st, we see a drop in salinity that persists for a couple of days, along with a continuous repeated drop in dissolved oxygen down towards basically unmeasurable levels. And at the very bottom graph here in this case is a bunch of gape data from mussels. And notice that those mussels close up around the middle of March 30th, and they remain closed all the way until the middle of, what is that, April 3rd. And so they are just toughing it out, holding their breath essentially that whole time during these low oxygen, low salinity river flow events. Same period of time, same data, but here I'm just showing you data for oysters, which are a little bit different in some ways, in that our oysters seem to be a little more adventurous and that they occasionally are opening up and sort of testing the water and then closing back down. But again, from March 30th to April 3rd, they're essentially closed, not going to be doing much feeding during that entire time period. Some data from 2025, again, showing a period of river flow in the middle of our graph at the top. And down below in salinity and oxygen, I want you to notice again these sort of daily drops in salinity, daily drops in dissolved oxygen. In some cases, that dissolved oxygen minimum is associated with a drop in salinity, but in some cases, the salinity is high and the oxygen is still low, which again implies something about the water that's either flowing through from the river or is stagnating in this area. In terms of gape, we see group closures. Everybody's closing up for hours and hours each day, and then generally reopening during high tide periods where, again, ocean water is pushing in. And during those closures, we see these drops in heart rate as well. These animals are trying to conserve energy. And then zoom in a little bit more on a different period of time. Here at our top graph, this scale of river flow is much, much smaller, but I want you to sort of pick out the day-night cycle in freshwater flow coming down the river here, which is going to be tied to basically the urban output. And associated with that, we are seeing these continued daily anoxic events and daily closures for all of these animals. And that repeated daily closure period of time for these animals didn't yield much in terms of successful survival. All of our little skulls here represent oysters or mussels that died during the portion of our deployment back in 2024. We lost all of our animals at the surface. We didn't lose any bottom animals because they were sitting in sort of a different body of water, essentially just being a few meters deeper in the channel. 2025 was also just as bad. We lost all of our surface animals over the course of really just 2 weeks, 3 weeks from deployment. And then our bottom animals, again, were faring a little bit better, but we lost most of our bottom animals over the course of about 2 months. So survival in Tijuana River is really, really poor right now. Kristen always cautions me to not end with a whole bunch of nasty, doom and gloom. So I'm going to try to sort of just talk about some of the challenges and sort of successes that we've seen out of this project. Things we've learned along the way: that mussels are a whole lot easier to work with than oysters when it comes to particularly measuring their heart rate. Measuring shell opening and closing is easy. It doesn't matter what species you use. We've used Magdalena here. We've done it on native oysters as well, and we've done it with Mytilus provincialis as their mussel. But those mussels are easier to find the heart rate. It's easier to find a good, strong heart rate, and over long periods, that heart rate signal tends to hold up. We've also seen lots of evidence that these animals do grow. And if you think about it, when we initially glue these sensors onto the animal, the magnet is about X, millimeters away from the Hall effect sensor. As that shell grows, as we move from left to right in our diagram here, the magnet, even when the shell is closed, is actually getting further and further from the Hall effect sensor. And that's manifested in this climbing minimum value that we see in our raw gape value. So I'm not expressing things as 0 to 100% here. This is sort of the raw voltage signal. And so what you're seeing here is this narrowing of that signal that we have to account for the fact that, yeah, the animal's growing, and so our baseline is constantly shifting. We definitely had to deal with a lot of sensor failures, and there are sort of physical waterproofing changes that we can make. The other thing is sort of on the hardware sensor end of things to basically handle individual sensors failing, handle that more gracefully. Because when we've got, in this case, 8 different animals feeding into one data logger, if one of their sensors shorts out, it tends to pull the entire data logger offline, and so we need to be able to handle that better. And so, just to sort of finish up, I do want to say that I feel like we've been reasonably successful in gathering a whole lot of long-term and pretty high frequency, high resolution data from these animals in a variety of different settings. And so it's been a reasonable demonstration that we can make these measurements, we can do them over long periods of time, we can broadcast that opening and closing data to the internet. We did it on a 15-minute interval. That's a choice that we can choose whatever time scale we want. But we were able to broadcast sort of real time group gape opening and closing data as we had set out to do. And so I feel like this is technology that is maturing. I'm hesitant to say it is fully mature, but we are sort of making strides there. So I want to just finish by acknowledging a lot of folks who pitched in on portions of this project, and of course our funding source from the NERR Science Collaborative project through the University of Michigan, and our folks who also sort of hosted and supported us, including San Diego Bay Aquaculture, the Los Penasquitos Lagoon Foundation, and California State Parks, U.S. Fish and Wildlife as well, who handle some of the permitting in down in Tijuana. And with that, I'm happy to go ahead and take questions.
Thanks, folks. We have a few minutes left to answer some questions, and we've got a pretty long list here, to be honest. So let's see how many we can get through. And if we don't cover them in session, I will pitch them to presenters to answer after, and we'll include it in the summary stuff if that sounds good for everybody. All right, so first question, does the estuary program have a sampling system that shows when the best time is to place clean shell? For instance, when the larvae are ready to set and then place the clean shell.
I think the answer is no at this point. That's something where we probably have to defer to the aquaculture folks who know a lot more about sort of when best times are to set. But yeah, in terms of these estuaries that we've been working in, I don't have a good sense for that.
I would also add here on the West Coast, there's a little bit of difference. So that would be really interesting for the native oyster. But the native oyster is generally not produced for aquaculture, commercial aquaculture production. And so most of our aquaculture is using the Pacific oyster from Japan. And so we don't actively harvest spat from shell in California. That does occur in Washington, but not here.
Thanks, folks. All right, next question. Is the BBC bivalve write-up identifying this protein source as climate-friendly a good reference? And there's a link included. If folks didn't see that one, I will paste it into the chat for everyone to see. But I don't actually know if you know anything about that. I have not seen this resource personally, but I guess
Yeah,
in a more general sense, is a climate-friendly resource.
I have seen this resource. It's pretty great overall. I think they have some ambitious goals for bivalve aquaculture listed in that. But overall, I would say it's a pretty reliable resource if you wanted to share it around with folks.
Okay. Next question. Is there much maintenance involved with the sensors? It looks like you cover the sensors in epoxy in addition to using super glue, maybe, to prevent them from falling off. I think you touched on this a bit towards the end, Luke, but is there anything else you might add about
Yeah,
maintenance?
so we encase the circuit board and the wire that's coming out of it in watertight or waterproof heat shrink, and then do a sort of layer of glue over the top of that to try and sort of provide the waterproofing. And then in terms of attaching to the shell, we're either using super glue initially or UV curing epoxy to get that just initial attachment on the shell, and then covering it with Z-Spar splash zone epoxy, that greenish stuff in some of the pictures, to provide the sort of main holding power and long duration. And we really have very few occasions where a sensor falls off of these things. Our failures tend to be often because wires end up getting cut or braided, and therefore we get water intrusion that way.
Okay, we are at time. There was a quick poll question that we were hoping folks could answer maybe on your way out. So Ariana, if you want to go ahead and launch that now, if it'll let you. And for the other questions, I will, like I said, we'll pitch them to our presenters and they can answer them, and I'll include that in the summary product at the end. But thank you so much to everyone for joining us today for the session. I personally learned a lot. This was super cool. As somebody noted earlier, this is really interesting tech. This is a really interesting use for Hall effect sensors that I have not seen before, so that's really cool. Um, if you have a minute when you're on your way out of the session after you answer the poll question, you'll automatically be prompted when you leave the session to take the exit survey. I do actually read the feedback, and it really does help inform how we, run the webinar series. So if you have feedback, please do put it there. You can also let presenters know what you thought of their presentation and like how you might use it as well in that exit survey. So all helpful stuff for all of us. Uh, Everyone, thank you again for joining us. The recording will be posted in the next day or so. So check back on our website or YouTube channel if you don't see it right away. It'll be posted, like I said, and it usually just takes me a day or so to process them. And thank you so much for joining us, and everyone enjoy the rest of your day. And thank you to our presenters. Great job.
Thanks, everyone. And thank you, Nick and Ariana.
Yeah, thank you,
I'll
everyone.
go ahead and end the poll here.
I can't wait to see.
I can share it now if you want, or I can just stick to results.
Sure. Let's see where
But
people landed.
I'll post it now if you want to see it.
Oh, there we go.
Nice.
Very nice.
I love your options.
Yeah.